Good morning. Welcome in the lecture Ontogenesis. It's the first time we meet, so please let me introduce. My name is Kamila Prochaskova, and we will spend this lecture and then the lecture in teratogenesis and seminar in teratogenesis together. It will be in the next semester. So it's the first time together today. What will be the topic of this lecture? We will go through these 6 chapters: ontogenesis, morphogenetic processes, developmental stages, control mechanisms of ontogenesis, signalization in early ontogenesis, and genes in early development. So, I have mentioned more times the word ontogenesis. What does this term mean? Do you know what ontogenesis is? And what's the difference between ontogenesis and phylogenesis, and between phylogenesis and evolution? Take your time to consider these terms. So, ontogenesis is the term describing individual development. On the contrary, phylogenesis is the development of species. You can draw the phylogenetic tree and illustrate this phylogenetic species development. Phylogenesis concerns whole taxons and is descriptive. On the contrary, evolution is the process behind phylogenesis, is the motor of phylogenesis. Evolution concerns the development of species or part of the body and is the motor of phylogenesis. So phylogenesis is the description, evolution is the process, and ontogenesis is the individual development. Today we will look at ontogenesis only. Ontogenesis is the term describing individual development. So the process, the sequence of temporally and spatially programmed morphological and functional processes leading to the change of zygota into the complex adult organism. If you look at embryos of different animal species, you can see here a frog, a chicken, a mouse, and the fish, zebrafish. You can see that the early embryogenesis is quite similar in more animal species. Oh, sorry. So you have seen it for a moment. So we discuss ontogenesis, individual development from zygota to an adult organism. How does the zygota know in which organism should it develop? So what is the program? What are the instructions? You have seen it for a moment, so you know it's DNA. So the genetic code. The genetic code, the DNA in the nucleus of zygota includes the instructions for the development. How does the genetic code look like, or the genes do like? They are coded by 4 nucleotides. You know it from biology and genetics, ACGT, and these genes and some non-genetic DNA code for the program of the development. But is it so similar? How can you imagine the process from DNA to the cat or another species? Do you think it is some process analogic to the computer program? So computers are coded in binary code. So do you think that this binary code, is a type of language, is similar to the genetic code, to the language of the DNA? So if you look at the code in the computer, it looks like a plan for a house, for example, here. In the code, in the program, there are all information describing every single tile in the house. So you can start from one end or another end, and in the end, you will have always the same house. It is really so in the development? Does DNA really work as the binary code in computer? It's not so easy, because in reality, in our genetic code, our DNA does not work as a map. No, these instructions are a little different and a little more complicated. We start from something. We do not know where will be every tile, every cell of our body. This is not coded in the genetic code from the beginning. But there are instructions how these cells will move to their final positions. So, not the house plan, but origami is the analogy for the development and the instructions in the DNA. During the development, the embryo is folded. You know from histology and embryology, folds, neural tube, all these foldings, defoldings are a little similar to origami. So this is the better analogy than computer binary code. And I will illustrate you this origami analogy in a specific publication concerning the turtle. In this publication, the authors searched for the source of the turtle of the turtle shield. You know that every turtle has his shield. It's on the dorsal side, and everything, all the body of the turtle is hidden under the shield. All legs, everything is under the shield. So, what is the origin of turtle sheet? The first hypothesis was that it can be the result of calcification of some skin plates. But later it was found that the turtle sheet is modified ribs. But you know, here's the picture of the human. You know that our ribs are inside the body, and our hands and scapula are on the dorsal side of the ribs. But in the turtle, scapula is placed under the shield, so under the ribs. How it is possible? Is the principle, and these authors describe it, and they have mentioned the principles of origami. It's the similar principle of folding, similar to origami. You see here, it's the human rib and muscle plate, and the human scapula. It's a normal position in human. Scapula is on the dorsal side of the rib. But in turtle, the proliferation and folding of the muscle plate will hide the scapula under the ribs, and this is the mechanism how the shield gets on completely on the dorsal side of the turtle. So folding, similar to origami. Now we will continue to the next chapter concerning morphogenetic processes. We can discriminate 4 morphogenetic processes: proliferation, distribution, integration, and reduction. And now we will go through all these 4 processes. The first one mentioned is proliferation. The base of proliferation is the cell division, and proliferation leads to the growth and shape differentiation of local cell populations. In the adult organisms, proliferation serves also to replacement of old and damaged elements. So proliferation is the first morphogenetic process. Proliferation starts in the stage of zygota, and you remember from embryology that the first cell divides into two cells, then every cell from these two cells divides into two cells. We have 4 cell stage, 8 cell stage. But is it really so proportional until adulthood? In the work of Bisotto, they have observed cell lines in the adult human brain, and they have supposed that the proportion from the first cell lines from the embryo will be similar. But they were surprised that the proportion between different lines from the first stages, from the first cells from the embryo, was quite disproportional. So we can discuss if it's due to proliferation, different proliferation between different cell lines, or due to, for example, reduction, next morphogenetic process. We will go to the next step then. The next morphogenetic process is called distribution or cell migration. What is the base of distribution? Distribution are shifts of single cells and cell populations from one place, from the place of their origin, to another place, to the place of organ formation. We can discriminate 2 types of distribution, passive and active. In passive distribution, you can see here some cell population, and this cell population will be moved to the place of organ formation by extrusion of the growth of other cells. These cells will exclude and between this cells population, and this cell population will be moved to another place. So this is passive type of distribution. And the second type of distribution is active, when the cell moves actively by polarization and then moves using microfilaments. The third morphogenetic process is called integration or cell association. If you imagine proliferating cells, if they are not integrated, we will have no final organism. If they are not tied together, they will flow away and would not form adult organism. So we need this morphogenetic process called integration. Integration is association of single cells into higher functional units by means of intercellular contacts. So integration between the cells and between the cells and intercellular matrix. The fourth and last morphogenetic process is called reduction or cell death, programmed cell death. Reduction serves to elimination of damaged and abandoned cells. It's common in adult too, but during ontogenesis, reduction serves also to the shape formation of developing organ by programmed cell death, and also for elimination of material useless for given species. I will show you picture to understand better what is mean with this sentence. So we have cell population here. In this cell population is homogeneous, and we need some shape formation here. A reduction is a way to the shape formation. If some of these cells will die with programmed cell death, the resting cells will form some specific shape in ontogenesis. So it's typical, for example, for some organ sculpting, for example, for fingers. Elimination of material uses for given species can be found in different species. For example, here in frog, the tail during development will be lost and the adult frog has no tail. Here in the picture, look at it please. You can see the leg of duck here and the chicken. And you know that there is a membrane between the fingers of the duck because it's the water bird. In the chicken, on the contrary, all fingers are completely separated. And this separation is, and the difference between this complete separation and this membrane is due to different portion of reduction, different portion of programmed cell death in the cells in this part of the body. So in the chicken, the reduction is stronger, is bigger than in the duck, and the fingers will be completely separated. In the duck, there will be a rest of some surviving cells, and these cells will form the membrane between the fingers in the duck. So cell death is a very important process during the whole life, but especially here in the prenatal development. So there are three main functions of cell death during ontogenesis. The first is histogenetic. Histogenetic function is not so important during ontogenesis. It's more important than in adulthood. Histogenetic function of the cell death is the elimination of damaged and redundant material. So it was the first one. The second, very important in human development, is the morphogenetic function, when the cell death defines the shape of structures. You have seen the picture with fingers. And the third one is phylogenetic, when the cell death reduces transitory structures like the tail in the frog. Cell death can be called also apoptosis. And you know that apoptosis is the topic of a special presentation of Professor Maresh, so you will hear more and more about apoptosis later. So now we will leave morphogenetic processes and we will go on to developmental stages. Which are developmental stages in human ontogenesis? The first developmental stage is cleavage. It's the first stage from fertilization to morula. Cleavage from the first cell to morula, to the stage of undifferentiated totipotent cells. Second stage is the stage of pattern formation. During this stage, from this undifferentiated cells in the globe of morula, layers will be formed. Germ layers, ectoderm, mesoderm, and endoderm will be formed. So this is the pattern formation, the first pattern of the embryo, basic body plan. And the body axis are defined during this developmental stage. Craniocaudal and dorso-ventral X are established during this stage. Next developmental stage is morphogenesis. Starts with gastrulation and follows then with organ shape formation. From gastrulation to organ shape formation. So morphogenesis, organ formation. Next stage, differentiation. During this developmental stage, cells are specialized for their functional and morphological specialization in the organs. And the last developmental stage then is growth, the enlargement of the size of shapes defined before. If we look to zygota, we have said before that the instructions for the development are in the genome. In the genome, we can find the genetic parts of DNA called developmental program. And this developmental program can be divided into two groups. These parts of DNA are involved in the development. So the first group of developmental program is the basic developmental program. This part of DNA codes for common metabolic and reproductive functions of cells. And this basic developmental program is functional during the whole morphogenesis. On the contrary, specialized developmental program, this part of DNA, of group of genes, codes for RNA and proteins, which are responsible for functions of differentiated cells. And this specialized developmental program is repressed during cleavage. And then its particular parts are induced, while the other parts remain to be repressed. So this specialized developmental program switches on and switches off in different stages of the development of the embryo. Not only genetic mechanisms are involved in the development. So in zygota, also epigenetic mechanisms are involved. Epigenetic mechanisms, you will hear more of epigenetics in biology. Epigenetic mechanisms influence the phenotype of organisms, because we know that the phenotype of organism results from a chain of interactions between cells population, and these epigenetic mechanisms influence the expression of genes and are not based directly on the genetic base. Epigenetic mechanisms switch the genes and switch their stages of expression and silence, methylation, usually methylation or other silencing. During ontogenesis, in a precise temporal sequence, particular genes are switched on in certain cell populations. It's called induction, while the other genes are switched off. It's called repression. And this switching is induced by epigenetic mechanisms. Back to embryos and back to, you remember the first stages, first developmental stages, cleavage. Embryos can be divided into two groups, mosaic or determinative and regulative embryos. What is the difference between these two types of embryos? In regulative embryos, all cells in the embryo remain for a very long time totipotent. So if you take one cell from the embryo, also in later stages of development, the embryo will develop normally because other cells will replace the function of the missing cell. So the cell determination occurs later during development. And the single cell of the early regulative embryo may give rise to the whole embryo. On the contrary, mosaic embryos, in mosaic embryos, the cell determination occurs early, already in the egg. And the cells of the embryo are differentiated very early, so the loss of a single cell may lead to the absence of the part of the embryo and cannot be replaced by other cells because the cells are not totipotent at the moment. They differentiate more early. Garden and his colleagues tried some experiments aimed to transfer of nuclei from differentiated cells to oocyte. And they tried if the differentiated cell can be changed to a totipotent cell. So they have taken some completely differentiated cells from the intestine of the frog, and they transferred these cells into enucleated egg. This enucleation has been done by UV ultraviolet irradiation. After a transfer of the nucleus from the differentiated cells into enucleated egg, they have observed that the complete embryo and complete adult frog can develop. So they have proven that completely differentiated cell, the nucleus of completely differentiated cell, can be dedifferentiated by some influences in the egg. So the differentiated cells can be dedifferentiated under very specific conditions. Gardner has received the Nobel Prize in Physiology or Medicine in 2012 for this discovery that mature cells can be reprogrammed to become pluripotent. Gardner and for the same thing also Shinya Yamanaka. Next chapter will be control mechanisms of ontogenesis. Control mechanisms of ontogenesis can be observed on intracellular level and extracellular level. We will go through these levels of regulation and control. So first, we will go to some regulations. In 1935, Hans Spemann and Hilde Mangold received the Nobel Prize in Physiology or Medicine for the discovery of the effect now known as embryonic induction, but they didn't know how it works. They have discovered an influence exercised by various parts of the embryo that directs the development of groups of cells into particular tissues and organs. They have transferred some cells from the donor embryo to the ventral side of host embryo, and then they observed that from these cells a new embryo developed, and these two embryos were sorry. So this and the orientation of the second embryo was like that. They were ventral side to ventral side oriented, and they supposed some influences they have called them organizers, which can induce the direction of development, but they didn't know what these organizers are. It was found later. Now back to intracellular regulations. On the intracellular level, the regulation of protein production can be on 4 levels. The first level is the regulation of DNA replication, then regulation of transcription, post-transcriptional regulations, and post-translational regulations. On the level of DNA replication, the first mechanism is gene amplification. Multiplication of particular genes can increase the proportion of transcribed mRNA and then subsequently the production of proteins. So if the gene is amplified, then these amplified copies can be expressed in higher amount, and this protein can be expressed in higher amount in the cell. Regulation on the level of DNA transcription is also differential gene expression. Different genes are expressed in different frequencies and amounts, and it's specific for particular cell populations at a concrete place and time. What is the mechanism of this differential gene expression? For the first time, histones. Histones are responsible for unspecific repression of DNA template activity. And then non-histone proteins. The non-histone proteins are specific, and they are responsible for specific activation of DNA template of specific region in specific time. They are called transcriptional factors. Histones are linked to the condensation of chromatin sections when the eukaryotic inhibition of gene expression remains in cell memory and is passed on to the next cell generations. So you know it very well from histology: heterochromatin and euchromatin. Heterochromatin can be constitutive, always folded to heterochromatin, and facultative, which can be changed and defolded to euchromatin. Specific situation on the DNA level is lionization. You know that women carry 2 X chromosomes and men have 1 X chromosome only in every cell. Y chromosome is very small, so women would have an advantage of the duplicated gene dose on the X chromosome, and it would be not ideal. So in female cells, 1 X chromosome is always inactivated, and this inactivation is random. So it's absolutely random in which cell which X chromosome will be inactivated. And this lionization compensates the gene dose between women and men. You know from biology that genes are inherited, or from Mendel laws, you know that genes are inherited, and it is not important if they come from the mother or from the father. But there are some exceptions. And these exceptions are called genomic imprinting. What is imprinting? It's the different activity of the same gene depending upon its origin from the father or from the mother. How is it possible? Normally, the genome of both parents is necessary for the normal development because some genes are imprinted. Imprinting means usually methylation. In the egg and the zygote, all genes from the father and from the mother should be demethylated, so it should be tabula rasa. But some genes remain methylated, and these genes has a stamp of the father or of the mother, and they act then differentially in the embryo if they are from the mother side or from the father side. In experiments, you can try to form embryos including maternal genomes or paternal genomes only. If you form genogenetic embryos missing father's genome, then you will observe normal development of the embryo but abnormal development of extraembryonic structures. On the contrary, if you form androgenetic embryos missing maternal genome, it will lead to the interruption of embryo development and normal development of extraembryonic structures. So you see that we need one copy from the mother and one copy from the father. Because during germ cell maturation, genes required for yolk sac and placenta development are inactivated in maternal genome, while genes controlling embryo development are inactivated in paternal genome. This process of methylation is reversible, so genes can be demethylated, but there are some specific genes that stay methylated in embryo. And you will hear then more in human genetics about some typical syndromes linked to some specific genomic imprinting. Now we go to the next level of intracellular regulation, post-transcriptional regulation. The post-transcriptional regulation can be formed by processing of cytoplasmatic mRNA precursors, which can be modified by splicing. You know that exons and introns in young pre-mRNA can be spliced, and you can splice introns only, or you can splice out also some exons from the pre-mRNA. And you get different alternatively spliced mRNAs, and these mRNAs then form different protein isoforms. So splicing is a very important mechanism leading to the diversity of proteins rising from the same DNA gene. Next, post-transcriptional regulation is a selection of specific mRNA molecules, which are then translated predominantly, and then regulation of ribosomes and mRNA interactions. Both these mechanisms can lead to some preference of some mRNAs and the formation of preferred protein. On the level of post-translational regulations, you can regulate protein amount during protein folding, during protein degradation, because proteins can survive for different time in the cell, and then they are degraded by proteasome, and the timing can be different for different proteins. What is very important in intracellular regulations are transcriptional factors. What are transcriptional factors? You will hear more from Professor Maresh. Transcriptional factors activate or block transcription of other genes by binding to their control region, to the promoter of some genes. Transcription factors play the key role in the development because transcription factors are responsible for the switching on and off of the genes during the ontogenesis. And now we will go to the next chapter concerning signalization in early ontogenesis, because we were now in controlling of the development on the intracellular level, and now we go to the intercellular level, to signalization. Signalization can be formed by direct contact of cells, direct receptors, or gap junctions, you know more from histology, or using a mediator, some chemicals carrying the signal as a second messenger, to the receptor, and then from the receptor the signal will be transferred by second messenger to the nucleus. The signalization between cells which are not in their direct contact can be autocrine signaling. If a single cell is signaling, then one cell will receive only a very weak signal. But in a group of identically signaling cells, each cell will receive a strong signal. For long distance, we can discriminate two main types of signalization: endocrine and synaptic. Endocrine signaling is transferred by hormones produced from one place and carried in the blood to target cells. Synaptic signaling for long distance is represented by neuron and the axon of the neuron. For a long distance, the signal from the neural tissue will be transferred to the target cell. Now, two important terms. Competence. What is competence? It is related to signaling. Competence is the cell ability to respond to an inducing signal. So not every cell will respond to every signal. The response requires a presence of receptors, specific receptors, then the presence of transport mechanisms and transcription factors. So only competent cell will respond to the signal. And the next important term is position information. What is position information? Position information determines the cell identity. The position information is important for the cell because the cell needs to know where this cell is located to act like the cell differentiated for the organ it is located in. Position information depends upon the genetic constitution and developmental history of the cell and provides the basis for pattern formation. I will show you in detail how the cell gets the position information. It can be demonstrated by the model of French flag. If you look at the French flag, you see three compartments of colors blue, white, and red. If you use this model for some cell population, we have here a cell population, you need to inform these cells about their position, and then they get the information of their position, and they will develop into the blue, white, and red cells. The position information is carried by morphogene. What is morphogene? It's a very important substance in ontogenesis. Morphogenes is a substance active in pattern formation on the basis of its gradient that provides positional value to the cells. In the model of the French flag, we have these undifferentiated cells. Every of these cells has the potential to develop as blue, white, or red. But the information given by a morphogene will say or inform the cell in which direction it should develop. So the position of each cell is defined by the concentration of morphogene. If the morphogene is produced somewhere here, the concentration will decrease by the distance from the source of the morphogene. So this decreasing concentration will inform the cells about their position, because here the threshold of enough high morphogene concentration will give the position information to the cells to develop in the blue direction. The positional value is interpreted by the cells, which differentiate them to form a pattern. So enough high concentration of this morphogene will give the information, the positional information, to develop in the blue direction. Then the lower threshold, the lower amount, will give the information to the cells to develop in the white direction. And then where the concentration of the morphogene is so low that it is not enough to give the signal for blue or white, the rest of the cells will develop in the red cells, in the last differentiation direction. Next very important mechanism in ontogenesis is lateral inhibition. It's a specific mechanism by which the cells inhibit adjacent cells from developing in a similar way to themselves. So the cell gives a signal to the neighborhood that all the cells in the neighborhood cannot develop in the same type as this cell. But the concentration of the signal decreases by the distance from the cell. So here the concentration of the signal will be so low that this cell can already develop in the same direction. Lateral inhibition is responsible for the development of a structure with typical regular pattern like that. For example, hair cells or some neurons and so on. The cell can get positional specification also independent of the environment. And it can be due to asymmetric distribution and asymmetric cell division, which leads to different daughter cells. So now we will go through the general strategy of pattern formation. The first stage is formation of the basic body plan, establishment of the body axis, craniocaudal and dorso-ventral. And second, then gradual formation of details by means of intercellular inductive interactions to morphogenes. You will hear more later. Cells diversify and their arrangement depends on position signaling between the cells and the cell program that modifies the response of cell to these signals in dependence of time. So we will go to the last chapter today, genes in early development. You hear some molecular aspects of development also from Professor Maresh. So something you have heard a lot of from him. So we will go only shortly through the genes, but we will look in detail on the principles of these early stages development. Genes involved in development can be divided into two groups, maternal and zygotic. Maternal genes are the genes of the mother, not of the fetus or not of the embryo. Maternal genes are expressed in maternal organism during oogenesis, and they control production of mRNAs and proteins deposited in the egg, as well as their distribution along the axis of the animal and vegetal pole. So maternal genes are responsible for the start of pattern formation. Maternal, not zygotic. Maternal genes contribute to establishment of body axis and to formation of basic body plan. They are the first genes involved in the development, and after the influence of maternal genes, it is switched to the influence of zygotic genes. Zygotic genes are present directly in the fertilized egg, in the zygota, and they are expressed in the embryo itself. So these are own genes of the embryo. But the development, early stages of development and pattern formation start by maternal genes. And then the embryo develops, remember the start of the lecture, like origami, so folding and growing and proliferation and reduction and other morphogenetic processes leading to the definitive body shape. To show you how the early development works, we will use model organisms, because biologists use also model organisms. The most common model organisms for ontogenesis are Drosophila melanogaster and Caenorhabditis elegans. We will work today with Drosophila. Drosophila is a small fly, you know it probably from your household, a very small fly. And you see here the cycle of Drosophila from fertilized egg to adult Drosophila. You see the first stages of formation, of axis formation, then segmentation, pupa, metamorphosis, and adult fly. Drosophila will serve for us now as a model organism, and I will show you the influence of maternal and then zygotic genes in early development. So, in Drosophila, maternal genes play the key role in the determination of craniocaudal axis. The first one is a protein bicoid, called bicoid. Bicoid defines the anterior part of the craniocaudal axis on the basis of anterior-posterior gradient. So bicoid protein, maternal protein bicoid, is a morphogene. The concentration is high in the cranial part of the embryo, and the concentration decreases to the caudal part. So it's a morphogene and gives a positional information to the cells in the embryo, and remember the French flag. Concentration of morphogene gives the positional information. So the first one is the bicoid protein and gives the information about cranial part. Next, maternal genes and their products, protein products, are hunchback and caudal. The concentration of hunchback mRNA is constant in the whole Drosophila, but in the caudal part, hunchback is inhibited by the protein nanos, and this inhibition leads to the decrease of concentration of hunchback in caudal part of Drosophila, and this morphogene will give some positional information to the cells of Drosophila, and serves also as bicoid to the formation of craniocaudal axis. So the craniocaudal axis in Drosophila is determined by proteins bicoid, hunchback, caudal, and nanos. You remember hunchback, you remember bicoid. The concentration of bicoid is the highest in the cranial part of Drosophila. Then hunchback, which is inhibited by nanos, so the concentration decreases from the cranial to caudal part of Drosophila. And the protein caudal, it's similar mechanism as in hunchback. Caudal is inhibited by bicoid, so the concentration of caudal protein is the highest in the caudal part and the lowest one in the cranial part. So these proteins define the cranial caudal axis in Drosophila. And all of them are morphogenes. The determination of cranial caudal axis is then a little more complicated. There are more proteins involved in the definition of the axis, for example, torso and other proteins. Torso specificates the terminal structures of the embryo. And now to the dorso-ventral axis. Dorso-ventral axis is defined by more products, more proteins. Protein dorsal, which is influenced by the receptor toll. Dorsal protein concentration is the same in the cytoplasm of all the cells under the vitelline membrane, but in the ventral part, the receptor toll is activated by the protein Spätzle, and the activation of the toll receptor by the ligand Spätzle leads to the determination or to the different concentration of the protein dorsal in different cells in the Drosophila, and leads to the determination of dorso-ventral axis. So it's also the morphogene. And the concentration of the protein dorsal in cells here under vitelline membrane is a morphogene, leading to the positional information in these cells and determination of the dorso-ventral axis. So after this axis formation, it leads to the differentiation of specification of these cells in different parts of the developing Drosophila, and then this cell population in the future will give the base for germ layers due to morphogene dorsal concentration. So once more, we have here hunchback. First, the craniocaudal axis is formed and dorso-ventral. Then it activates gap genes. Gap genes are switched on by the genes of the primary differentiation. The gradient of bicoid protein initiates the expression of zygotic hunchback gene. Hunchback protein then plays a key role in the switching on the expression of other gap genes. Here you can see some gap genes, for example, cripple, giant, and others. And regional distribution of different transcription factors give rise to a number of unique sectors. So, from the axis differentiation and gap genes, we go to the activation of per-rule genes, and these per-rule genes initiate the body segmentation. So, once more, maternal genes are responsible for the axis determination, and then we go to zygotic genes from here, gap genes and per-rule genes, which are switched on by products of maternal genes, and zygotic genes are responsible for body segmentation. They switch on then segment polarity genes and homeotic genes. You have heard a lot of homeotic genes from Professor Maresch. Per-rule genes are switched on by concentration of gap genes and maternal genes. You see here that the concentration of bicoid, giant, hunchback, cripple switches on the expression of specific per-rule gene in specific region of the Drosophila. So here, for example, if type 2 is switched on by this specific concrete concentration of hunchback, concrete concentration of bicoid, and this concrete concentration gives the positional information to these cells, and these cells will express this per-rule gene if type 2. There are more per-rule genes, for example, hairy, even skipped, primary and secondary. They are they form parasegments in the Drosophila. Each of these genes is expressed in a series of stripes along embryo with a periodicity. This periodicity corresponds to alternate parasegments on the basis of local concentration of gap gene transcriptional factors. So, this periodic segmentation along craniocaudal axis leads then to formation of segments and parasegments and compartments. Segments are defined by segment polarity genes. Segment polarity genes are activated in their response to expression of per-rule genes. What is their function? First, the function is a specification of the structure of particular parasegments, because we have here these parasegments. And second, determination of the fate of cells in each parasegment through the cell signaling. So the segment polarity genes form the borders of parasegments and segments and parasegments, and the cells in one segment then will have the same fate together. So I will mention then the word compartment a little later. So this is the illustration of the influence of these segment polarity genes. You can see some names of the genes, wingless or hedgehog, in Drosophila. In human, there are some analogs of these genes called sonic hedgehog and VNT genes. You have heard these names from Professor Maresch. And they are responsible for borders formation of these parasegments and segments. And now we have one segment due to position information given by different concentration of the products of these genes. And now we go to the important term in ontogenesis, compartment. What is compartment? Compartment is a discrete area of embryo containing exclusively the descendants of a small group of founder cells present at the time of compartment patterning. And compartment delimits boundaries of cell lineages, and all cells within compartment are under the common genetic control and share the same fate. I have mentioned it in the description of segment and parasegment formation. They share the same fate and then cannot migrate between segments, and then they cannot cross the boundary to become a part of another compartment. So in one compartment, all the cells will share the same fate. Next important group of genes responsible for the early development are homeotic or selector genes, Hox genes. They specify the identity of individual segments. So we have the segmentation, thanks to the previous genes, and homeotic genes give the information of the future fate of the individual segment. So these Hox genes not say but inform the cells in compartments, then they will develop into the antennapedia or a bit in different parts of the Drosophila body. So they select different segments for different future parts of the body. In other organisms, in human too, we have analogic homeotic genes as in Drosophila. They are not completely same, but very similar. The principles are always similar. So Drosophila is a very good model organism because the principles of early development is very similar to the early development of all organisms. In 1995, the Nobel Prize in Physiology and or Medicine was given to Edward Lewis, Christiane Nüsslein-Volhard, and Eric Wieschaus for the discovery that mature cells can be reprogrammed to become pluripotent. It's similar as the experiments of Cardan later, so it was the similar work a little, and they have shown many informations that I have shown you now about signalization in early development. It was Drosophila, model organism, but how does it work in human? A human is a mammal, so it is not easy to explore mechanisms of early embryonic development in human because of technical and ethical problems. So we have some model organisms more related to human than Drosophila, for example, mouse and bovine. Here in the table there are some genes involved in early development in mouse and bovine, and the function is very similar as maternal and zygotic genes in Drosophila development, but it's mouse and bovine. But in this study, the authors compared these mouse developmental genes to the human ones, and they have shown that the principles are really always very similar. That the development is in the beginning influenced by maternal genes, and then later it switches on to the zygotic genes. Here you can see mouse and mouse in red and human in pink, that maternal RNAs are the first influence in the early embryonic development. So here are some human genes of early development, some examples, and they are compared to other species, and you can see that these genes responsible for early development are quite conserved and quite similar in all species. So we don't know enough about early human development because it is not so easy, but it's very intensively studied, and we know that the principles of switching on, switching off morphogenes and so on are very, very similar in human as in model organism Drosophila. And it was the story about normal development. And in the next semester, we will see in the lecture and practical teratogenesis, and we will discuss what happens when the development goes wrong. So I thank you for your attendance, and see you in the next semester.